Good morning. So today we are taking a very important topic in the area of RDN technology. So as far as the RDN technology is concerned, it is called as recombinant DNA technology. Uh, Basically deals with the fusion of two different DNA molecules. And if we join two different DNA molecules with the help of some specific enzymes called as T40 ligase or ET ligase, it creates recombinant DNA. That is recombinant DNA molecule. So, but in order to assess the properties of recombinant molecule, it has to be transformed into the host. And if we're taking host as a bacteria, it is called as bacterial transformation. So it's a process by which DNA can be transformed into the bacteria. But it happens only during the rapid growth, that is called as log phase of the bacterial cell. So as you can see on the screen, it represents the E. coli cell having some specific adhesion zones. So basically, DNA transformation is a process by which DNA can be transformed with the help of foreign recombinant DNA molecule in the bacteria. So, as you can see here on the screen, that this particular membrane, it consists of lipid molecules, and these lipid molecules, they have high mobility, high moments because of their electrostatic nature. So these kind of negatively charged phosphate molecules makes it difficult to enter the RNA molecule into the cells. So, in order to easy those steps, there are some specific processes. So you can see here that these resin zones are physically enough, but by virtue of having that negatively charged phosphate molecules, it repels DNA molecules to enter into the bacterial cells. But it can be made easier with the help of using some specific solutions. And you can see here that as we add DNA, by virtue of having that negatively charged, it gets spread. But it can be made easier with the help of using some particular solution. Solution is being called as calcium chloride solution. This calcium chloride, it imparts basically a positive charge. It interacts with the negative charge, either with the lipid molecules on the other side, the DNA molecules, and it creates electrostatically neutral situation. And under this neutral situation, these calcium chloride molecules, they get interacts with lipid molecules, I can see here, as well as the DNA molecules. So makes some neutral kind of situation. So once we load on the temperature, initial temperature is very high, maybe room temperature, but once we load the temperature to 4 degree, so it congeals the lipid membrane, stabilizing negative charge phosphates and making them easier to sheet. So under this condition, DNA ultimately get transformed under the presence of this ionic shield into the bacterial cell through adhesion joints, and this process is called as bacterial transformation. And in this process, there is one peculiar step, it is called heat shock at 42 degrees centigrade. So after the addition of calcium chloride, if we impose heat shock at 42, it creates temperature imbalance on the other side of the bacterial membrane. It sets off the current. So within this ionic shield, DNA can be set through the adhesion